We'll take a look at active transport in bacterial cells in this lecture. In active transport, substances are going to be moving from areas of low concentration up the gradient to where there's higher concentration. So we're going to be going up the gradient, and by going uphill, you do need to use energy to do active transport. The type of molecules that are going to be moved across the membrane by active transport, we are going to be using a carrier protein, also called a pump. And because we're using a transport protein, we're going to be transporting things that are hydrophilic across the membrane. And these would be your larger polar molecules. So for example, glucose or amino acids, and then also ions would also be moved by active transport. There's two different types of active transport that bacterial cells can do. They can do primary active transport, where energy is going to be directly used and all of the molecules are going to be moving up the gradient. And then secondary active transport, energy is going to be used indirectly. So you'll notice that one of the molecules will be moving down the gradient while the other is going to be going up its gradient. In primary active transport, the molecules are going to be moving up the gradient. So these green molecules are going to be moved from low concentration to high concentration inside the cell. And we're going to be using energy directly here. So you can see some ATP here in the picture being used to power this primary active transport. In secondary active transport, energy is going to be used indirectly. And in this picture, the bottom, that's secondary active transport. And you can recognize secondary active transport because you have one molecule. Here we have the hydrogen ions moving from high concentration to low concentration. So you have one molecule going down the gradient, and then the other molecule is going to go up its gradient. So sucrose will go from low to high concentration. We're going to be using energy indirectly because we had to use energy over here on the top to establish our hydrogen ion concentration gradient to allow secondary active transport to work. So up here on top, this is an example of primary active transport. So notice that the hydrogen ions are moving from low to high concentration, so they're going up the gradient, and we're directly using energy, some ATP, to do that. So this allows the hydrogen ions to be higher outside the cell than inside the cell, and this is what allows the bottom picture, the secondary active transport, to work. And just some other terms that go along with secondary active transport, we have Symport and Antiport. So Sym means together, and this is just saying that both of the molecules are going to be transported in the same direction. So they're both, for example, going into the cell, and that's what we have here. On this picture on the bottom, this would be an example of secondary active transport or Symport. It's also called co-transport, so you might have learned that term as well. Antiport, anti meaning the opposite. So in this one, we're going to have one molecule going in the cell and then the other will be going out. So they're going in opposite directions, and that would be antiport. It's still secondary active transport, but it's just describing how one molecule is moving into the cell and the other is going out of the cell. Group translocation, this is new, so you haven't learned about this one before. This is only done in bacteria and archaea. So eukaryotic cells cannot do group translocation. It does require energy, so it is a form of active transport. And what happens is as the molecules moved into the cell, it's going to be chemically modified. So maybe it's going to be phosphorylated, so we'll stick a phosphate group on it. The best known translocation system is the phosphoenolpyruvate sugar phosphotransferase system, which you do not have to know that name. Okay, just know what group translocation is and be able to describe what happens. So let's take a look at a picture of group translocation. In group translocation, we're going to be moving molecules from an area of low concentration to an area of high concentration. And in this example, we're going to be moving some sugars across. So if we take a look at the glucose on the bottom, you can see that as it's moved across the cell membrane by the transport protein, it's going to become chemically modified and it's actually going to be phosphorylated. So as the glucose moves across the membrane, we're going to phosphorylate it. So this is going to require energy because we're moving up the gradient, and where the energy is actually coming from, which you don't need to know because you don't see any ATP in the picture. But remember those phosphate groups, those chemical bonds between them have a lot of energy in them. So we're just going to take a phosphate group off of basically a pyruvate molecule that has a phosphate group on it. And so we'll just take the phosphate group off there, and then that will phosphorylate the glucose as it moves in, and that's what supplies the energy to do group translocation.